Okay, well let's start then.
Yeah. Hi everyone. Good morning, good afternoon, wherever you are around the world. So today we've got a really interesting paper being presented by Masa, which is on the real-time luminescence assay for cytoplasmic cargo delivery of EVs from Osaka University. From sort of my reading of this paper, it sounds really, really interesting. So I don't think it needs any more introduction from me. Masa, over to you.
Okay, thanks, Will, for introduction. And I am Masaharu Somiya from Osaka University in Japan. And it's already late in Japan. It's 11 p.m., so I will try my best not to fall asleep during this talk. And my talk will be focusing on the novel assay to study a cytoplasmic cargo delivery of EVs. So I think I don't need any detailed description of EVs because everyone is familiar with Vesicle. But this talk, I will focusing on the mammalian cell-derived EVs. And EVs are secreted by cells and probably deliver cargo, proteins or nucleic acid into recipient cells. And lots of people believe in that EVs possibly deliver cargo to other cells, but honestly, I'm not so much, believing this hypothesis because there is a very few evidence of solid evidence of the cargo delivery from donor cells to recipient cells, and based on this assumption, a lot of papers are coming out these days. And if you search exosomes and drug delivery on PubMed, you can see clear trend of increase of publication like this. So people are expecting that EVs are a good tool to deliver a biological material to recipient cells. But as I mentioned before, only a small direct evidence of cargo delivery has shown so far. And this figure is from 2020 science paper. And people believe that exosomes or extracellular vesicles are internalized into cells by endocytosis. And after that, they release their cargo into cytoplasm by unknown mechanisms. But few papers show that evKylo delivery process might be inefficient. I will talk about this point later, but some papers clearly showed that the process is not efficient as we expected. And the problem is that cargo delivery process is still unclear. Now we have some methods to study the cargo delivery process and the biological function of EVs. Yes, we have a fluorescence labeling technique or immunostaining to study endocytosis process or intracellular localization of EVs. In addition, we have some tools to study the functional output or phenotypic change of recipient cells, like reporter assays or some bioassays, etc. However, we still don't have any good tools to study cargo release process. Because of that, we still don't know whether EVs are actually releasing their cargo into cytoplasm. If you think about the viruses, they have viral fusion proteins that enable them to deliver their cargo. In the case of SARS-CoV-2, they have spike protein on their surface. The spike protein binds to the cellular receptor and then internals into cells by cytosis. And after that, the spike protein fuse with target membranes and start releasing their genome into cytoplasm. As shown here, membrane fusion is the key step for viral cargo release especially viral fusion protein is essential for establishing viral infection. On the other hand, we still don't know whether EVs have fusogenic protein or fusogenic activity. I think this is the fundamental question of EV cargo delivery hypothesis, but we still don't have any good tool to study this process or fusogenic activity of EVs. This is a general virology experiment. You have a virus here, and you put the virus into the host cell. And after several days, you can measure the viral amplification in the host cell, like qPCR or something like that. So in virology, quantification of virus particles or viral titer is relatively easy. And of course, functional output, in this case, amplification of viral genome in the host cell can be easily measured. And of course, this output is specific for virus genome, and the assay is quantitative. On the other hand, we have EVs here. In the cargo delivery assay, we put EVs into recipient cells, and after like 24 hours or 48 hours, we can measure the functional activity of EVs in recipient cells. However, quantitative assessment of EV cargo delivery is not feasible. And sometimes readouts can be easily interfered by some experimental artifacts or contaminant EV fraction. And unfortunately, functional assay is not always quantitative. So this is my motivation of this work. Quantitative functional assay is the key to study and understand the function of EV. Functional assay should directly measure the amount of cargo in cytoplasm recipient cells. And also functional assay should generate specific readouts upon cargo delivery in recipient cells. So based on these assumptions, we try to establish the functional real-time assay to study the cargo delivery. We named this assay as EV cargo delivery assay, evCD assay for short. The idea of this assay is very simple. We use a split nanoluciferase, which is composed of two components. First one is high bit tag, which is 11 amino acids, very small tag. Another one is large bit protein, which has 18 kilodaltons. And their components have very high affinity, like 0.6, 0.7 nanomolar. When they complement each other, they become enzymatically active, and they can emit a luminescence signal. So the idea is very simple. We put a cargo protein with the hybrid tag, and we chose EPN01 as a cargo protein. I will talk about this later. But the idea is very simple. When we put EVs with hybrid tag as cargo protein to recipient cell that is expressing a large bit, after the internalization, if EVs release their cargo into cytoplasm, the large bit protein and high bit tag will complement, and we can see the luminescence in the recipient cell. And by using this assay, we can assess the kinetics and efficiency of cargo delivery into recipient cells. In this assay, we use virus-derived fusion protein, Vsvg, as positive control because, as you know, Vsvg is a potent fusogenic protein from virus. So if EV has this kind of protein, they must be functionally deliver their cargo into cytoplasm. So I will explain about EPN01 as evKylo. This protein is designed by computer, and this protein was first reported in 2016 Nature paper. The protein is composed of 3 domains. First one is membrane binding M domain. Second one is interaction I domain. And the last one is late budding L domain. The key component of this protein is I domain, because this domain drives to form a nanocage, which has a 60 protein monomer. And the late budding domain is also important to recruit this nanocage into escorted pathway. So after the expression of this protein in mammalian cells, this nanocage are recruited to escorted pathway and released from cells encapsulated inside EV. This is a cryo-TEM image of EPN01 containing EVs. In this image, you can see a lot of nanocages inside a single EV. And according to this paper, on average, about 14 nanocages are encapsulated in single EV. In this study, original MIC tag was replaced to high-bit tag to be used for EVCD assay. So we first characterized the high-bit tag cargo protein in donor cells. We transfected high-bit tag EPN01 expression plasmid into 293 T cells. And after the lysis of transfected cells, we detected a clear band here showing that EPN01 can be expressed in 293 T cells very efficiently. And to confirm this EPN01 nanocage are encapsulated into the EV, we perform the co-immunoprecipitation experiment. In this experiment, we precipitated EVs by using EV-specific anti-CD81 monoclonal antibodies or anti-Vsvg antibodies that capture the EV. If the high-bit tag cargo protein are encapsulated inside EVs, we should immunoprecipitate these EVs with high-bit tags by using these antibodies. After the incubation of donor saps and supernatant with antibodies and magnetic protein GBs, after the extensive wash, we measure the amount of high-bit protein in the precipitate. As shown here, compared to mouse IgG control, we can see that the clear enrichment of high-bit tagged proteins in precipitate by using anti-CD81 antibodies and anti-Vsvg antibodies. In addition, after the disruption of EV by detergent, we saw a dramatic decrease of high-bit signals, suggesting that high-bit tagged EPN01 proteins are actually encapsulated inside EVs, and after disruption, these cargo can be released to outside of the vesicle. So we purify the high-bit tagged protein cargo encapsulated EVs by using PEG precipitation because of the ease of this method and relatively high throughput method. So after the collection of supernatant, after the centrifugation, we incubated the supernatant with PEG overnight, and after the precipitation, we resuspended this EV pellet in PBS. And we characterized the PEG precipitated EV by Western blotting. As you can see here, all the common EV markers, CD9, CD63, or CD81 was detected by Western blotting. And of course, after the expression of Vsvg, we can see that the Vsvg protein was actually on the EV fraction. So now we have two kinds of vesicles. EV with EPN01 hybrid tag, this is the EV containing a hybrid tag EPN01 nanocage. We don't know whether this vesicle without physiogenic protein is capable to deliver cargo into cytoplasm. And now we have another vesicle here. This vesicle has hybrid tag EBM01 nanocage with Vsvg on their surface. This vesicle must have a fusiogenic activity, and this vesicle must deliver cargo into cytoplasm of recipient cell. So here is the actual evCD assay. The experiment setting is very simple. We transfected donor cells with EPN01 expressing plasmid DNA, and after the collection and precipitation, we purified the vesicles and applied to the recipient 293 T cells. Of course, this recipient cells is expressing large big protein. So in this experiment, we typically use up to 100 femtomole total hybrid per well. And this experiment was done in the using by 96 well white well plate. And we measure the luminescence signals in recipient cell by using microplate reader for up to 90 minutes. Again, this assay is very simple. If we have a cytoplasmically released cargo proteins, we can see the luminescence signal. Okay, this is the first experiment of evCD assay. When you put EVs without Vsvg fusiogenic protein, we couldn't see any increase of luminescence signal in recipient cell. First, we saw this result. I'm kind of surprised because there's no cargo release in recipient cell. On the other hand, in the presence of Vsvg protein on the surface of the EV, we saw a clear increase of luminescence signal in recipient cell. So I think Vsvg is essential to deliver cargo proteins into recipient cell. And without this fusiogenic protein, this EVs have a very weak activity of delivering cargo into recipient cell. So I guess some people wonder other purification method is working in this assay. So we check whether ultracentrifugation method is working in this assay. So we transfected cells, we transfected donor cells with plasmid DNA, and after the collection of vesicles, we purify the vesicles by the ultracentrifugation, and we apply the vesicles into the recipient cell. And here we have a similar result as previously shown. You can see that in the presence of Vsvg, you can see the clear increase of luminescence signal in recipient cell. On the other hand, without Vsvg protein, we couldn't see any increase of luminescence signal in recipient cell. So you may wonder, what is the effect of incubation time? So we incubated cells with vesicles for 24 hours and measure the luminescence signals for recipient cell. As you can see here, as shown in previous slides, there is a clear increase of luminescence signal in recipient cells treated with EV with Vsvg. But without Vsvg, we couldn't see any increase of signal compared to the negative control. So I think some of you may consider about the detection limit of this assay. And based on our calculation, evCD assay is enough sensitive to detect very small amount of high beam. This is our calculation. As I mentioned before, EPN01 nanocage has 60 monomer of protein. That means single nanocage has 60 high bit tags. And on average, 14 EPN nanocage can be encapsulated inside single AV. So this is a very simple calculation. 60 times 14 equals about 800 high bit tag per EV. On the other hand, we have a recipient cell that is expressing large bits. So we measure the luminescence signal of this recipient cell mixed with the high bit peptide, which has a known concentration. And you can see here a clear linear curve here, and the luminescence signal was dose dependent of high bit peptide. So maybe we can detect less than 0.1 femtomole high bit in like 100,000 recipient cells. This is equivalent to 600 high bits per cell can be detected in this assay. So if you compare these two values, I think evCD assay can detect only a single evKylo release can be detected by this assay. So based on this calculation, we couldn't see any increase of luminescence signals in recipient cell, suggesting that EVs without fusiogenic protein have very weak or no activity of fusion, membrane fusion. And previous studies also found that the evKylo delivery efficiency is quite low. In this current 2020 Nature Communication paper, they use CRISPR-Cas9 single-guide RNA reporter system. And based on this system, they found that only 0.2% of recipient cells can be activated by the evKylo. Another two studies using Cre messenger RNA or Cre protein. And in this study, they found that only 6% of recipient cells become reporter gene positive cells. By using Cre protein, they found that only 1% of recipient cells are reporter gene positive. So here's my first conclusion. EV-mediated cargo protein delivery is inefficient process. Based on our own EVCD assay, we can detect a very small amount of cargo delivery event per cell. And without Vsvg, we couldn't see any significant signal in recipient cells. And other three studies shown that very few recipient cells can be reporter gene positive. So I think delivery efficiency is generally low, but I should emphasize that delivery efficiency might vary by lots of factors, such as sensitivity of assay and cell types and amount of EVs applying to recipient cells. So these biological contexts must be taken into account. But I think generally speaking, EV-mediated cargo delivery is not so much efficient. So the next part, I will explain about the application of EVCD assays to study the delivery mechanisms of EVs. First part, I will talk about fusion kinetics and the mechanisms of EVs by using Vsvg protein and fusion deficient mutant of Vsvg. Second part, I will talk about the investigation of endocytosis inhibitors and membrane fusion inhibitors by using evCD assays. The last part, I will explain about real-time imaging of evKylo delivery. So as I mentioned before, Vsvg is essential for cargo delivery of EV. And here you can see the picture of membrane fusion mechanisms of Vsvg. Here you have two membranes, viral membranes and host cell membrane. And after the acidification of endosomes or lysosomes, the Vsvg conformation of Vsvg can be changed. And after the insertion of fusion loops to the target membrane, they become trimerized. And after the conformation under change, these membranes can be fused. And if we put some mutation in here, chlorine 127, we can see the dramatic decrease of fusiogenic activity of Vsvg. Here you have wild type Vsvg proteins. They have clear, potent fusiogenic activity in pH 6. In contrast, if you have a mutation here, chlorine 127, you can see a very low fusiogenic activity in a lower pH. So we apply this mutation to Vsvg proteins, and we purified the EVs with Vsvg with or without mutation. Here you can see a clear difference between a wild type Vsvg and mutant Vsvg. This result suggested that EVCD assay can be useful to decipher fusiogenic activity of these kind of fusiogenic proteins or intrinsic fusiogenic activity of natural EVs. Here we have another example of the EVCD assay. We can study the effect of inhibitors on cargo delivery by using Csvg EV. In this experiment, we use 3 types of endocytosis inhibitors, including chloropromazine, which is for clustering-mediated endocytosis. Dinosaur is the inhibitor for dynamine, that is essential for endocytosis process. EAPA is the inhibitor for macropinocytosis, which is one type of endocytosis. And we have 2 kinds of membrane fusion inhibitors. Chloroquine is known to increase the pH of endosomes and then block the membrane fusion of viral proteins. The Pheomycin A1 is the proton pump inhibitor that is also known to increase endosomal pH. By using these 3 endocytosis inhibitors, we can see that the clear decrease of cargo delivery efficiency in recipient cells. And also membrane fusion inhibitors that increase endosomal pH clearly showed that the decrease of cargo delivery efficiency in recipient cells. 1 micromolar of chloroquine is not enough to suppress the cargo delivery, but 50 micromolar is enough to suppress, completely suppress the cargo delivery. In contrast to the chloroquine, Pheomycin A1 is very potent that can suppress the cargo delivery as low as 10 nanomolar, and 100 nanomolar of Pheomycin A1 completely block the cytoplasmic cargo delivery. So this is the last data of my talk. As I mentioned before, we can detect the luminescence signals from recipient cells. So we try to capture the real-time kinetics of cargo delivery by using imaging techniques. Luminescence microscope was commercially available from Olympus company, and this machine LV200 was used to capture the real-time cargo delivery of EVs in recipient cells. In this experiment, we used EPN01 containing vesicles with Vsvg protein. Here you can see the previous data showing that the Vsvg vesicles, Vsvg positive vesicles, can increase the luminescence signal in recipient cells as fast as 10 to 15 minutes after the application. So here is the imaging data. As you can see here, after the 15 or 20 minutes, you can see the bright dots here, here, and here, and here. Okay, play again. After the application of EVs to recipient cells, after 20 minutes, you can see the bright dot here, 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 and here. Okay, I think this is the first demonstration of direct observation of cargo delivery in recipient cell. So I think this is pretty much useful to decipher cargo delivery kinetics and intracellular localization of cargo in the recipient cell. So here's the summary of evCD assay. The assay is very sensitive that can detect a very small amount of hybrid protein in recipient cell. And in this assay, physiogenic protein such as Vsvg is essential for efficient cytoplasmic delivery. And without Vsvg, we couldn't see any cargo delivery in recipient cell. And luminescence imaging reveal the intracellular trafficking of EVs in real time. So here's my second conclusion. Based on our results, the delivery mechanisms of EVs can be assessed by our novel evCD assay in real time. And I think EV-based DDS can be efficient if EVs are modified with physiogenic proteins or other delivery mechanisms. Otherwise, I think EV-based DDS without fusion activity is not useful and is not applicable in a clinical setting. And I believe that further investigation is needed to decipher detailed mechanisms and efficiency of evKylo delivery. As I mentioned before, biological context is important to draw a conclusion whether evKylo delivery is physiologically relevant or not. So I hope a lot of people use this assay to really understand the efficiency and the mechanism of evKylo delivery. And material used in this work will be available, and expression vectors are already deposited in Adhesivity. So there was some technical issues in Adhesivity, but I hope these plasmids will be available soon, hopefully coming week. And synthetic peptides, sequence of these peptides is already available in some papers, as well as our recent preprint uploaded in October. So maybe you can find our articles, and please try this assay in your own system or in your own cell type. And before closing my talk, I want to introduce some famous quotation from Sidney Brenner. Progress in science depends on new techniques, new discoveries, and new ideas, probably in that order. And I hope our technique, evCD assay, will help to understand the evKylo delivery mechanism. And I hope this assay will lead to the new discoveries and the new ideas in this community. So I'd like to thank my collaborators in Osaka University, especially Professor Nagai and Professor Hatsui. They helped me a lot to conduct a luminescence imaging. I'd like to thank my lab members and colleagues in the Kuroda Laboratory in Osaka University. And I'd like to thank my funding agency, JSCS, the JGCS Scholarship Foundation, and MEX. And thank you for your attention, and I'd be happy to take any questions.
Thank you for that, Masa. That was a really, really fantastic talk and really interesting. It's generated quite a lot of questions, so I think we'll start from the beginning. Oh, hang on. So we've got a question from Alan Ezrin. Do you want to unmute?
Fascinating talk, and thank you if you can hear me. Just wanted to talk about the binding protein and to solicit your opinion, some of the historical work on the phosphatidylserine flip on, we'll say, malignant exosomes as far as exosome binding with an avidity. Any role of annexin in the surface of exosome playing that role of the spike protein, so to speak, for actually adhesivity? Any thoughts on that?
I'm not sure about activity of luminescence, and I would say some protein can be fusiogenic in the context of cargo delivery, but I think it is really important to identify some specific protein that is responsible for cargo delivery activity of EVs. And I hope someone discover some specific protein that is responsible for a fusiogenic activity of EVs.
And then I'll go on to my next question, which goes to the essence of the work as far as the definition of cargo. You talk about a detergent lysis as if you're assessing the intervesicular material as cargo. And do you know that the detergent actually can lyse these? Most of the time we believe that these microvesicles are relatively inert, need very high quantities of things like guanidinium to lyse them. Might you be looking at a detergent dispersion and actually what you're calling cargo just be contaminant bound between the vesicles as aggregates as opposed to true cargo? Do you have any data that suggests that you're actually lysing the vesicles with some type of marker to say that the lipid vesicles are not intact? Because I don't believe a detergent like Triton will actually lyse these vesicles. Your thoughts on that, please?
Sure. I think, yeah, we use 1% of Triton X to lyse the vesicles. And I believe that that concentration is enough to completely lyse the vesicles. And previous work published in 2016 clearly showed that the EPN01 cargo is sensitive in the proteinase by after the lysis of Triton X. So I believe that at least EPN01 cargo encapsulating EVs can be completely lysed by the Triton X or some another detergent.
That's lovely. And sticking with the Triton, we've got a question from Emmeline.
Yeah, hello, and thank you for your really interesting talk. In my lab, we are using the full-length nanoluciferase, and we realized that some chemical compound, and mostly Triton actually, have some effect of on the nanoluc activity itself. Like in my hand, 1x Triton is 1% Triton is increasing nanoluc activity. So I was wondering if you try to check the different chemical in your experiment if they affect the enzyme activity itself, or if you didn't check.
You mean a detergent can interfere the luciferase activity, right?
Yeah. I mean, in my hand, yeah, at least that the detergent affect the activity itself. And I know, I mean, I have some colleagues that experiment the same kind of trouble shooting with some other chemical compound on, I mean, it was the full length one, once again, it was not the split one. So I was just wondering if it happens to you too to have this kind of issue, or if you check.
I haven't tried the detailed studies to whether detergent actually affect the activity of nanoluciferase, but I believe that the full-length nanoluciferase is pretty much stable and is not interfered by chemical compounds or detergents. So I think general settings like 1% Triton X do not significantly affect the activity of nanoluciferase. That's my guess.
Okay, thank you.
Okay, and we've now got a question from Suenya.
Yeah, hi. Very nice talk, Masa. I have a quick question about the Vsvg. So Vsvg is known to use for the pseudotyping lentivirus for receptor-independent endocytosis. So if you do Vsvg on the EVs, you have more uptake without receptors. So I wonder how you normalize the uptake efficiency between Vsvg positive and negative EVs in your experiment.
That's right. I guess presence of Vsvg can significantly increase the uptake, not the delivery. So we couldn't, unfortunately, we couldn't normalize the uptake efficiency in the ADCD assay. So Yeah, you're right. Uh, Uptake can be simply enhanced by Vsvg protein.
Yes. Hello. Thank you, Masa. Very nice talk. I have a question about, as you know, many people have shown, many research groups have shown that beneficial effects following MSC EV administration in vivo. And how would you explain these effects, when the uptake of EV cargo is so low? Any thoughts on this?
Yeah, I believe some EVs are capable to deliver their cargo in different biological settings or biological contexts. So I couldn't provide any solid evidence of that, but maybe some type of cells can secrete fusion-capable vesicles, and that could be biological active in some settings. But I'm not sure about any factors that is totally responsible for cargo delivery.
Okay, thank you.
Can I sort of follow that up, Masa, with a bit more of a question? When you showed that really nice video at the end of your presentation, I think there was sort of 5 or 6 of the cells you could track the cargo in them. Do you think that you could add, if you increased the number of EVs that you were applying, you would see it in more of the cells, or it would be a more concentrated sort of uptake in those specific cells?
Yeah, I saw dose-dependent signals in the 293 cell only. Maybe another cell type is more sensitive to the vesicles, or another cell will not be sensitive as 293 cells, but I'm not sure about other cell types.
Okay. And we've got a question from James as well on this same topic. James, if you want to unmute.
Yes, I just was, and you sort of got at this, but have you tried actual like antibodies against the Vsvg receptor, which I'm not even sure what it is, but.
Yeah, that's a good question, but we haven't tried. And actually, we have antibodies that neutralize Vsvg, but in this assay, we have to use a large amount of antibodies, so we couldn't try that experiment, but that's interesting.
Okay, and I had a sort of follow-up later, I'll just ask now is, are there any quote unquote native fusogenic proteins that you could try that would not be as immunogenic perhaps as a viral protein?
I think I want to try some kind of native fusogenic protein such as syncytin-1, which is encoded by human genome. So it could be less immunogenic, but I'm not sure about that. But I think we should definitely try that.
Yeah, sorry, I have some problem. Okay, so my question is regarding the isolation method. Like I studied for a long period liposome, and we use the PEG for shielding the surface particles of the particles. So my question is, if you use the PEG for isolating the EVs, aren't you shielding the part with the protein and the lipids which are on the surface on the vesicles? And that could be a reason why your uptake is so low, because you're shielding what actually needs to be used for the uptake. Have you tried another isolation method except from the PEG?
Yes, we tried ultracentrifugation method to isolate the vesicles, and it worked. And after the ultracentrifugation purification, we applied the EVs to the recipient cells, and we saw a similar result as PEG precipitation. So, as you mentioned before, it is true that PEG precipitation has some disadvantages to purify pure EVs. But I think in this assay, PEG precipitation is not a problem to get a functional readout.
Hi, thank you. That was a very nice talk. I'm wondering if afflomycin affects the ability of the cells to take up EVs indirectly by its effect on inhibiting autophagy. So is there an inhibitor that can stop this uptake of EVs without affecting other cellular processes?
That's a good question. And I know that vancomycin has another effect on the cellular function like autophagy. And yeah, I'm not familiar with that kind of autophagy thing, but Yeah, I'm not sure. I'm not really sure, but yeah, and another treatment like ammonium treatment can increase the pH in lysosomes or endosomes. That could be a better method to block the fusion by increasing a pH in endosome or lysosome. But I'm not really sure about another method.
Okay, I've now got another question from Richard, if you want to unmute.
Yes, I had a question about your imaging, your lifestyle imaging, because Vsvg assays usually take a lot of time to do to get an exposure onto in a live setting. How long did you expose to get lifestyle images?
You mean luminescence imaging?
Yeah. Yeah.
The exposure.
Yeah,
exposure time is about 60 seconds per image. So it's relatively long to capture a real-time event. But it's a problem of the sensitivity of nanoluminescence. So it's in best settings, we need 60 seconds to capture the single image. And that's one of the problems in the assay.
And your Vsvg packing in the producer cells, do you know that whether the Vsvg affects the packing of EVs in the producer cells? So if maybe the amount of
possible, because yeah, expression of Vsvg in donor cells can be toxic sometimes. So it could affect the loading efficiency of cargo into EVs. But I'm not really sure about that point. But that's an important question.
Hi, thank you for a nice talk. I was just wondering, maybe I missed it, but did you try putting the HIBIT tag, I guess you did, on the luminal side of the EVs on a membrane protein or a luminal sort of non-membrane associated protein? And why did you choose this cage? Is this just for increasing the amounts of the HIBIT tag, or what would you say to that?
Yes, that's a good question. Actually, we tried tetraspawning to tag HIBIT on their N-terminal, and we put these data on the preprint, so you can find that data on the preprint. And actually, as you mentioned before, the nanocage is the very good tool to count the HIBIT tag, because they have 60 HIBIT tags in single nanocage. It can be a significantly accumulated. So it's very sensitive in the imaging and counting. So that's why we use EPN01 nanocage in our assay. But definitely you can try tetraspawning with HIBIT in the assay.
Did you try a non-membrane associated EV protein?
No, not yet.
Okay. Do you think it would take longer for the cage to kind of unpack, or does it even need to be unpacked for the interaction between the split nanoluciferase? In other terms, do you think that the tag would be trapped inside this nano cage, or did you?
Yeah, that's
check?
a good question. I'm not sure. But I hope the HIBIT tag is exposed on the surface of the nanocage, because we can easily detect the HIBIT tag in our assay. But I'm not really sure. That's a good question. Yeah.
Thank you.
Thank you.
Thank you. I think that is all the questions we've got. There are some comments in the chat if people want to look back over them, but I should also apologise for anyone's names that I've horrendously mispronounced. Apologies for that, but if there are no more questions, I think we could all say a massive well done to Masa. A very, very well written paper, very well presented and well conducted throughout the whole presentation. So well done.
Thank you.